So Watson and Crick, they noticed that specific base pairing that they're able to come up with upon completion of their model suggested a possible copying mechanism for the genetic material. Since the two strands of DNA are complementary, each strand will then act as a template for building a new strand in replication. So what that means is if these two strands can come apart, well, now you have half of a molecule that is predetermined. And what that means is since we have the law of complementary base pairing and we have Chargaff's rule, we can then understand how these letters here are now going to fit in to make a second strand. Now, on the other side, we can have what we'll find here on the left side of the strand that's separated. So we have the complementary base pairs for each one. And if these two now come together, we have two new identical strands. So it takes the parent molecule, this one here, it's required to unwind, which means break down the center where we found all those hydrogen bonds that we talked about last time. And then two new daughter strands will be built from the parent strands. Now, at this time, there were a couple of competing models, which we'll look at in just a second. But if we take a look at this diagram here, you can see how the parent separating produces these open ends here for the new nucleotides, the new nitrogenous bases to connect chemically. And now we have two, we could say half new DNA strands. So let's talk about those competing models just for a second, and it will kind of bring the whole thing together. One of the first types of models that were brought forth was the conservative model, where the parent was able to produce a copy of itself, but then the parent came back together after producing the new daughter strand. Well, through a number of complicated experiments, this was disproven. This was not the case, so conservative model is a big no. There was also the case of a dispersive model that some scientists believe was happening, where in a dispersive model we have your original parent strands breaking up into a number of pieces that you can see with these dark blue colors, and they would go ahead and build new fragments represented by the light blue colors and then kind of piece back together. So each new strand was kind of a mix of old and new. And again, more experimentation was done, and the dispersal model, again, was shown to be incorrect. So Meselson and Stahl, they do a very unique experiment, which you can read about in your textbook, where they're able to show that instead it is this middle model that winds up winning, and it's called the semi-conservative model. And what it says is that with each new strand, half of the new strand is going to come from the parent and half is going to be brand new. So in this case here, the only way, and this goes with the last two slides that we looked at, the only way this can happen is if these two strands come apart from each other, they separate, and then each one acts as a template for the new strand, which is this light blue color here, would be the new strand. Now, both of these wind up being daughter strands, but they contain part of the parent, which makes it not conservative like this one, but semi-conservative, because half is old and half is new. And if you were to continue on with your replications, you would see that the old strand is conserved in still another generation, and that the new generations then would bud off of your new strand that you built in the last generation. So in that way, it's semi-conservative, where we keep a parent around a parent strand for the next generation. In this picture here, the thing I wanted to highlight the most was the appearance of what we have as a 5' and a 3' end, which you can see up here. And you'll notice that the left side is 5' and the right side has 3'. Now, if you follow this 5' down, where we get to is right here. So the directionality of each half of, in this case, the double helix, we have a 5' to 3' direction. But how can that be if this is 3'? Well, the starting point for that one would actually be down here. Let's imagine that this is the end of a particular strand, and let's track backwards and see where it goes. So here, 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 and we reach the 5'. So this one is actually kind of starting here and ending here. Now, is there a true start or end? Not really, because DNA is anti-parallel, which means that one sugar phosphate backbone goes in one direction, this way, and the other sugar phosphate backbone goes in the other direction, this way. Now, this will have some consequences in how DNA is copied, and we'll take a look at that whole big series of events next. Okay, so this is just a partial picture of some of the ongoings during DNA replication. Some things to note here are some things that stick out to us is that the only real piece of double helical DNA, as we know the double helix, is right here. The rest of this, as you can see, is split. So this right here is kind of like the beginning of the Y fork. And we see some new players that we haven't seen before, such as this enzyme called helicase. We see another one called primase that seems to be doing something. DNA polymerase, which is this big blob here. We see a primer, an Okazaki fragment, leading strand, and lagging strand. Now, there's a lot going on in DNA replication, so we're going to slow it way down, and we can take a look at what each of these players has to bring to this process. Before we start zeroing in on those, I do want to just mention that the copying of the DNA when it goes down is remarkable in its speed and accuracy. 12 to 15 different enzymes are in play during the major process of copying it. Here, this picture is even more complicated because there's even more players than you saw than just one slide ago. So again, we'll go through and we'll take a look at each one of these and find out what their function is. Okay, so first things first, we always have to have a starting point, and that starting point is going to be called the origin of replication. Not too creative, but it gets down to the bottom line of where it happens at. Okay, you have to have a starting point. And how it's going to wind up happening is you're going to wind up with a little bubble. A bubble is going to start off this replication process. Now, in contrasting a prokaryotic cell to a eukaryotic cell, prokaryotic cell has a lot less DNA, and it's found in a ring. So that is only going to need one origin of replication, or one replication bubble. However, we are eukaryotes, so we have a lot more DNA in number than a prokaryote does. So for us to get this done in an efficient process, we're going to have a number of those replication bubbles up. So eukaryotes have anywhere from hundreds to thousands of origins of replication that open during this DNA replication process. Here, if we take a look at these two pictures, you can see what happens in a prokaryote or a bacterium. So right here would be the initial beginning point. This is your first bubble, your first origin of replication, and it's opening up. And when it does open up, I kind of drew over it, but if you check out really close in here, when the bubble happens, you now have single strands with their nitrogenous bases sticking out, and you have the same thing going on here. The light green color, this right here, represents new nitrogenous bases, new nucleotides that are being brought in by an enzyme called DNA polymerase and attached to their proper complement, their matching pair. So that process is going to continue. It's going to work to either side, since this DNA is circular, and essentially build a whole new volume of DNA identical to the first one. And when it's complete, this will peel off and it will form a brand new DNA molecule inside of the same bacterium. So you're going to have two copies, which means that this bacterium is probably ready to split at this point due to binary fission. Down here, we have the eukaryotic DNA replication. Here you can see a couple of replication bubbles opening up. Again, these are the free nucleotides, but the greens have been brought in by DNA polymerase to connect to those. And these replication forks in a eukaryote are going to go in both directions. And what that means is that these bubbles, this left one and this right one, are going to combine somewhere in this region in the middle. And when they do, you got this nice bigger bubble here, but it will continue to replicate to the right and to the left until I have two brand new strands, two daughter strands with half the strand still a parent and half the strand brand new. At the end of each replication bubble, and we're focusing in now at more players and more key happenings, we're going to find a replication fork, which is a Y-shaped structure. Now, that Y-shaped structure is right here. Here is where the Y begins. Okay, so this is the very end of the left side of a replication bubble. And that is where you're going to have the Y becoming longer, or I should say wider, due to a specific number one player who is your helicase, which is this green blob right here. This green blob is working down the center of the DNA molecule, splitting it into two single strands. Here would be single strand 1, and here would be single strand 2. And what's going to happen, of course, are these are going to serve as templates for when new nucleotides are going to be added to each one of these prongs that is sticking out, which is just a nucleotide from the parent strand. Now, one thing that you can do, well, let's just say that helicase goes down, opening up the replication bubble further. Now, that replication bubble can collapse back on itself unless it's held open, and that is the job of a single strand binding protein, which we see right here and right here. So it's like two helping hands that are holding open that bubble so the rest of the enzymes can go to work on it and keep it open. It doesn't recollapse and bind back to each other. Next is an enzyme called topoisomerase, which you can see down in the corner right here. It's this big blue blob. And topoisomerase, the best way to think of its action is if you have a lanyard and you go ahead and you twist the lanyard, okay? Put a few twists in a lanyard. I'm going to use my finger as helicase. I'm going to push helicase down, opening up the double-stranded helix. But what I see happening is it gets real tightly wound down at the bottom. Okay, the same thing's going to happen with a DNA molecule. If this is a double helix and I go down like this, it's too tight and the helicase will stop in its tracks. So what's going to happen is topoisomerase is going to come in and it's going to snip it, unwind it, swivel it, and rejoin it so the helicase can keep on doing its job. The last enzyme that I'm going to mention here is this one here called Primase. Now, the copying enzyme, which we'll talk about in just a minute, it needs a short segment called a primer laid down in order to begin where it's going to do its copying. Okay, so we have a primase enzyme laying down not DNA but an RNA primer. And this is not going to be too big of a primer. It's actually going to be kind of small. And let's take a look at some of the details on it in our next slide. Okay, so it's called Primase, and what it's going to lay down is an RNA primer, as we said before. So instead of A's, T's, G's, and C's, it's going to lay down A's, U's, G's, and C's. It can start the RNA chain from scratch, and it's going to add those nucleotides one at a time until it reaches about 5 to 10 of them. Then you have a proper starting point for the next big enzyme that's going to do most of the work. That enzyme is called DNA polymerase. It catalyzes the elongation of a new DNA strand at a replication fork. So basically, it's putting the right nucleotides in place on what's now a single strand because it's been opened up. So in this picture here, what we see is that the DNA polymerase is this green blob here and the green blob here. And notice that as the new strand is being built right here, it's nice and smooth. Okay, and it's just continuously adding its bases, no problem. But in the case over here, notice that there's not one arrow, but there's 1, 2, 3 arrows. So this is being created in a discontinuous type of way. It's going to be made of fragments, and that's what we're going to look at next. So we need to bring back into the picture the idea of anti-parallel elongation. So what's going to happen is that on one of the strands, and we know they run opposite to each other, you're going to have a continuous movement of that key enzyme that adds the nucleotides. And on the other side, we're going to have kind of a disjointed, fragmented way of adding it. So the enzyme that I was talking about that does add those nucleotides is called DNA polymerase. We haven't dropped its name yet, but here we mention it, it's synthesizing what's called a leading strand, and that's the one that adds it really smoothly. And let's take a look at how that happens. And actually, yeah, we'll start on this picture here. And look at this top just for a second. Here is your replication bubble. Here's your replication fork. And from the primer, the leading strand is built non-stop in a forward direction. So what we have here is we have helicase opening up this double strand of DNA. We have in the back an RNA primer, which is where this DNA polymerase 3 enzyme attached to. So it attached right here, and now it's working forward, adding new nucleotides to each exposed base. This one will be the next one, this one will be the next one. The grays, if you remember, are the single-strand binding proteins that hold open the DNA so it can do that. And it's just going to continue and continue until it reaches a place where the molecule is done being copied. Now, if we look even closer at it, what we're going to see is that it's moving forward in a non-stop, continuous fashion. And on the other side, we see what's called the lagging strand. Let's take a look at that next. The other strand is called the lagging strand, and that's the one that's built more in a discontinuous fashion. DNA polymerase must work in their direction away from the replication fork in order to accomplish this. The lagging strand is synthesized as a series of segments called Okazaki fragments, which are joined together by DNA ligase. All right, so those little pieces that are built separately from each other, those are your Okazaki fragments, and they're going to have little nicks in between them when all is said and done and their job is complete by DNA polymerase. So DNA ligase is another enzyme that's going to seal all those nicks to make the DNA right again. Okay, so here is a picture showing how these Okazaki fragments are built. And what we can see here is this is an RNA primer, and it's been laid down by Primase. And now DNA polymerase 3 is going to come in and attach to the 3' end, and it's going to start copying what we have here as a single strand. So here are the copies, or here are the nucleotides that are copying the single strand. And it's going to reach a point at where it will actually run into another RNA primer. And what it's going to do, it's actually going to dislodge from the strand that it's currently on, and it's going to go backwards to the other side. Because one thing I'll throw in here is that this particular copying process, believe it or not, overall is going to the left. So let's see what happens here in the next couple of steps. It reaches another primer on the DNA strand, and that's its signal to release. And what it's going to do is release, and it's going to keep going to the left until it finds a new 3' end, which it finds here. It's going to attach, and then it's going to copy to the right. So it will keep going to the right until it hits this one, and then it will dislodge again and go even further upstream. So although it's kind of going in a certain type of, like this type of motion here in pieces, overall, the directionality of the copying is to the left. And that's what we call discontinuous copying, or copying on the lagging strand. So a couple more enzymes we've got to throw in the mix here. How are these RNA primers removed? The answer is through this enzyme right here. This is called DNA polymerase 1. So we could say DNA poly 1. Compared to this one here, which was DNA polymerase 3. We'll just call it DNA poly 3. And this one here, its job is to remove this red piece. Because remember, DNA is DNA. It's not made of RNA. It has thymine instead of uracil. So it's going to come in, it's going to replace all the little red pieces, or the RNA primer. And the last thing is there'll be some nicks that need to be sealed up, and this enzyme right here, which we mentioned in the last slide, DNA ligase, is going to come in and it's going to seal the nicks and make this a nice and smooth, brand new DNA molecule. This table here is a great table. You can see that the proteins, the enzymes that are helping in the process are down here in the leftmost column, and their function is here on the right. I can't say enough as to how great this table is in terms of noting who the key players are when DNA gets copied and what their jobs are. So learn it well. Okay, so with copying anything, mistakes can be made. Now, 99.999%, everything is good. But unfortunately, every once in a while, a mistake does happen. So there needs to be a repairing mechanism. Sometimes it's called mismatch repair, but it needs to be proofread after those nucleotides are laid down to make sure that every letter is right. So a proofreading type of action takes place, and a number of enzymes are going to take care of that for the new DNA molecule. Why is there a need? Well, of course, DNA can be damaged by a lot of different things. As you can see here, chemicals, of course, radioactive emissions, so energy being put into DNA molecules, X-rays. You guys know that, when you go to the dentist, they throw a big lead vest on you because they don't want to irradiate the DNA in your chest and your vital organs. UV light. We know to limit our exposure to UV light so we don't set up conditions for skin cancer to occur. We also have mutagens in cigarette smoke. There's something like 400 carcinogens inside of any sample of cigarette smoke. So when a wrong letter is laid down, when a wrong nucleotide is put in place, we need nucleotide excision repair to happen. It needs to be excised or cut out. And that happens through a nuclease, another enzyme that's going to help out the DNA to be just right. So in this case here, we can see that something has happened. We'll say a person is laid out in the sun for too long, and that UV energy made what's called a thymine-thymine dimer up here in the DNA. And you can see that it is not supposed to be there. You have a big lump coming out of the DNA strand. So what do? You get a nuclease that comes in, and it's going to cut out that piece because that piece is not wanted. Then we have another polymerase come in, set up the right nucleotides in place, and after the other piece has been cut out. And lastly, to seal up the nicks that are there, DNA ligase will come in, make this nice and smooth, and your DNA molecule is fixed and ready to go. Eukaryotic chromosomal DNA molecules have at their ends nucleotide sequences called telomeres. They don't prevent shortening of the DNA molecules, but they do postpone erosion of genes near the ends. All right, so what do we have going on here? Well, at the end of a DNA, you have a whole bunch of sequences that repeat each other. They don't really code for anything. It's just like extra DNA. And the reason is every time DNA replicates, what we find out is that it gets shorter and shorter, and you'll see that in a second. So a telomeric portion of a DNA strand is the very end tip that's going to prevent the chiseling into the genes after a number of replications, which could eat up some genes that are pretty important. So by having all this extra DNA at the end, it's going to help keep that DNA healthy and keep those genes intact. Down at the bottom corner here, you can see at the ends of each one of these chromosomes, you have a couple of lights, okay? And those are the telomeric portions, so they're the end parts of the DNA molecules, those segments that repeat each other that don't really code for anything. All right, so here's the problem, okay? So when we have DNA polymerase go to work copying a DNA strand, what happens is when you get to somewhere like here, okay? In this case here, DNA polymerase has nothing to attach to. So after this one here is built and this RNA primer is removed, there's nothing really to help DNA polymerase lay down new nucleotides. So essentially what you did was you just shortened this molecule. So this is the end of the molecule, the very last part of it, which means that when it separates, when this strand here and this strand here separates, and the new nucleotides are laid down, it only goes this far, okay? Before, our DNA strand was this long, and the next time it replicates, it will be this long. So each time a DNA replication happens, it gets shorter and shorter. So having that extra DNA there is going to help out the process of keeping our genes safe. There is one type of cell, though, that we can't afford to shorten up, and that, of course, is a stem cell, because a stem cell has the unique capability of becoming any type of cell. And to chisel into those genes, to have those genes be eaten up, that means that every tissue, that every type of cell that receives that stem cell copy is not going to be quite right. It's going to be missing something. So we have an enzyme called telomerase that's going to take care of that. It's going to catalyze the lengthening of telomeres in stem cells. The shortening of telomeres might protect cells from cancerous growth by limiting the number of cell divisions. And there is evidence of telomerase activity in cancer cells, which of course may allow those cancer cells to persist and almost become like immortal. The last thing I wanted to talk about here was just the overall structure of DNA and what it does when it condenses in meiosis and mitosis. So here we have our double-stranded helix. We're all very familiar with how this looks. But when it comes time for replication, what we see is it starts wrapping itself in a very specific pattern around these weird looking proteins here, and together these are called histones. So there's a very unique pattern of wrapping around these histones, and you can see the histones seem to be wrapping themselves into something much bigger. When we see DNA wrap around histones in kind of like an alternate pattern like this, we call them nucleosomes, and they tend to look like beads on a string. But let's take a look at what this DNA grows into with the next slide. So here we have a histone DNA complex that is looping at this point into what's called the scaffold area, and you can see that the scaffold area is part of what will become the X that we all know and are aware of that we see in karyotypes. When we look in the microscope during cell division at prophase and metaphase, this is what we see. So it's amazing the DNA and protein combination that produces those big X that we know as the chromosomes. And maybe this picture also helps you understand why they couldn't quite figure out whether it was the protein or the DNA, which truly was the genetic material, because you can see that the DNA is very closely intertwined with that protein. And lastly, I just wanted to say that most of the chromatin is loosely packed in the nucleus during interphase. We said it's kind of just loose spaghetti just about everywhere. It condenses prior to mitosis, and when it is loosely packed, we call it euchromatin. Euchromatin is going to be pretty AT rich a lot of the times, adenine and thymine, and often that means that those areas are copied quite a bit. During interphase, a few regions of chromatin are highly condensed into heterochromatin, which tend to be a little bit more guanine and cytosine rich, because those had the 3 hydrogen bonds compared to the 2 hydrogen bonds of adenine and thymine. So if you have a very rich segment of G and C, it's going to be tougher to open it up and copy that area versus that of A's and T's. Now, are there any G's and C's in the AT region? Of course. But we just kind of look at how rich it is in the 2 versus the other 2, and to make a determination if that's a region that's likely copied or not during interphase. So I hope this talk helped you understand DNA replication a little bit better. Keep reading your book. Watch this a couple of times if you need to. You have the fill-in notes to go with this. So I'll talk to you next time.